Before we can discuss COPD, it is important to first understand how the human lung works. Gas exchange begins when you inhale and air goes down your windpipe into tubes in your lungs called bronchial tubes. Your bronchial tubes branch into thousands of smaller tubes called bronchioles. These tubes end in bunches of tiny round air sacs called alveoli. Small blood vessels called capillaries run through the walls of these air sacs. When air reaches the air sac, oxygen passes through the air sac wall into the blood in the capillary. At the same time, carbon dioxide moves from the capillary into the air sac. The airway and the air sac are both elastic, which means that when you inhale they will stretch out, and when you exhale they will deflate. COPD, or chronic obstructive pulmonary disease, is a progressive disease that can make it harder to breathe and can become worse over time. COPD symptoms include large amounts of mucus, wheezing, shortness of breath, chest tightness, just to name a few. In COPD, less air flows in and out of airways because of one or more of the following reasons. The airways and the air sacs lose their elastic quality. The walls between many of the air sacs can be destroyed. The walls of the airways become thick or inflamed. Or the airways can become thick with mucus and clogged. Cigarette smoking is the leading cause of COPD, with 80 to 90% of people who have COPD having been heavy smokers at one point in their life.
Genomic analysis is the identification, measurement, or comparison of genomic features such as DNA sequence, structural variation, gene expression, or regulatory and functional element annotation at a genomic scale. Geneticists wish to map the genes of all organisms in order to better understand the world we live in. The map information can then be used in identifying gene markers, helping regulate and splice specific genes, and ultimately help cure diseases. One of the main ways that genomic analysis is used today is through comparative genomic analysis. Scientists often compare human genomes to those of mice, fruit flies, and other organisms with extensively mapped genomes. This is possible because all organisms at their core share the same building blocks, DNA. For instance, 60% of the human genome has been conserved in the genes of the fruit fly. This similarity allows us to use fruit flies as small-scale models for research such as cancer testing. This image is an example of comparative genomic analysis. Here scientists were researching how proteins existed without stable states naturally. These proteins are called intrinsically disordered proteins. In this specific experiment, scientists were analyzing the population difference between intrinsically disordered proteins that had different functions in different organisms.
Since the genomic analysis is to identify, measure, or compare genomic features, there's a standard method to perform the genomic analysis. There are 4 basic procedures to conduct the analysis. First, identify a study population. To look for genes that are associated with a particular trait, a population must be chosen that uniquely fits a physical or behavioral model. Secondly, measure key characteristics of the members of the study population. Taking measurements helps to further define a population and to outline the specific traits and features they share. The next step is to look for genetic variants that differ between the case and control populations, so that we can compare and identify how features are genetically different. Lastly, make conclusion with all the information collected. The next slide shows how genetic factors affect COPD by using genomic analysis. According to the chart on the right, the research was taken by genomic analysis. The graph illustrates variation in COPD self-reported in the United States across race and gender. As shown on the right, the prevalence of COPD has generally been higher among whites and women. Although the data is limited to compare COPD in different racial ethnic groups, the available data suggests that differences in COPD may exist. Therefore, potential differences in COPD was found between racial ethnic groups, including genetic differences, by using genomic analysis.
In order to understand the present research, we must establish some context. The link between genetics and COPD first began with the discovery of alpha-1 antitrypsin in 1964. Lack of this protease inhibitor caused early onset COPD since ATT protects against lung tissue from inflammatory cells. A detailed mechanism can be found on the link below. Discovery of ATT, like many other early efforts at genetics, relied on family pedigrees, twin, or segregation studies. The sample sizes were limited and the progress was very slow. As the cost of sequencing became cheaper, methods such as meta-analysis, which looks for correlations in previously published genetic studies, began identifying more loci associated with COPD via everything from telomere shortening to epithelial repair. A comprehensive table can be found in the link below. However, large-scale genetics still had few issues. They were highly dependent on sample size, but not many high-quality databases had complete phenotypical data, so analytical depth was shallow. The present state of research is represented in a new paper published September of this year. This GWA attempts to resolve these issues by using the newly assembled UK biobank in conjunction with a custom SNP array designed to favor rare mutations. This nested control case study successfully identifies 6 novel genes associated with COPD by looking at the extremes of the phenotypes. Let's take a brief look at the methods they used. First, build a large databank complete with tissue samples and spirometry data. In this case, forced expiratory volume over 1 second. Next, filter samples with missing phenotype data, dubious smoking history, or FEV measurements that do not comply with standards. Next, select the remaining samples from the high, low, and average FEV from populations who have never smoked and heavy smokers. Then perform correlations of SNPs using custom arrays to identify any regions indicative of the case, COPD. The 3 major comparisons carried out are low FEV versus high FEV in non-smokers, low FEV versus high FEV in heavy smokers, and heavy smokers versus non-smokers. We will examine the Manhattan plots for each. In a Manhattan plot, the x-axis represents the location of the individual nucleotides, while the y-axis represents a logarithmic p-value. This particular plot shows genes that are related to COPD in non-smokers. The red dotted line indicates the cutoff for genome-wide significance, and the red data points show significantly associated genes. Previous findings that are confirmed in this study are labeled in bold, while those underlined are new findings. This next plot compares genes that may explain why some smokers are able to live out their habits while others are not. And this final plot shows candidates for genes that indicate a higher propensity towards smoking.
In conclusion, genomic analysis is still a young science, but it is quickly becoming an important tool for fighting diseases like COPD because it can help to identify the risk of such diseases before an individual ever contracts any symptoms for it.